Imagine a time when our ancestors weren't crafting tools or painting cave walls, but swinging through the canopy, more akin to apes than humans. It might seem like a lifetime ago, but this was just 7 million years back. Back then, the line between humans and apes was so blurred that it was nearly indistinguishable. Fast forward millions of years to today, and the tables have turned. Humans reign supreme, while our closest primate relatives, the great apes, seem almost primitive in comparison. From bigger brains to bipedalism, countless theories have been proposed to explain this dramatic divergence. But one theory stands out, a genetic enigma buried deep within our DNA chromosome 2. On this mysterious chromosome, unique to humans, holds the key to understanding our remarkable evolutionary journey. But what is chromosome 2? Why do we have it and apes don't? And most importantly, why does it matter? Today, as anyone who has gone to a biology class before will tell you, two things remain constant. The mitochondria are the powerhouse of the cell, and the cell is the building block of life. Taking things to a more advanced level, we begin to see just how intricate the cell really is and how much it shapes who we are. Besides being the building block of tissues and the smallest living unit of an organism, the cell has another nifty trick up its sleeve. Located in the nucleus of a cell in eukaryotic organisms, which include animals, plants, and fungi, the chromosome is an extremely important building block of who we are. Chromosomes decide what we all look like. Regardless of the animal species or type of living organism, the chromosome is a long thread-like structure made of DNA and proteins that carry genetic information. Found in every living organism, chromosomes carry the genetic information necessary for growth, development, and reproduction. Essentially, you get everything from skin color to height to even some diseases like diabetes, sickle cell anemia, and cancer, all from the information contained in your chromosomes. This is your genetic makeup, your fundamental biology, and what makes you, you. And before we go on, it's important to differentiate between chromosomes and DNA. Although used interchangeably sometimes, the two actually have a difference. While chromosomes carry the genetic information of an organism, DNA is the molecule that contains the genetic instructions essential for development. Think of a chromosome as a long, organized structure composed of DNA and proteins. Each chromosome contains a single continuous DNA molecule that carries many genes. In other words, DNA is the substance within chromosomes, and chromosomes are the organized form in which DNA is packaged. Now, something about human DNA is the similarities it bears to other animals. For example, we share approximately 85% of our DNA with mice, 60% with fruit flies, about 80% with cows, and about 90% with cats and dogs. However, among all animals, the ones we are closest to are chimpanzees, with whom we share about 98 to 99% of our DNA, making them our closest living relatives. This isn't surprising, though, because we both originate from a common ancestor. Although unknown, it is believed we split from the ape line of evolution after this common ancestor around 6 to 7 million years ago. But here is where things get really concerning. What exactly caused the split? What made us so different that we walked upright and built houses? What made us humans? Since the early 1990s, scientists have been aware of a special anomaly within the human genome. At the time, we weren't sure what exactly it was, but we did know something was different. Fast forward to the Human Genome Project, complete in 2003, where we mapped all human genes and finally found it. Let's see. The project not only mapped out the human genes, but also the genes of other animals. It was during the project that we discovered that our relatives like chimps, orangutans, bonobos, etc. had 24 pairs of chromosomes. Now, seeing as we all had a common ancestor, logic dictates that if chromosomes are hereditary, we should also have 24 chromosomes. But surprisingly, to the shock of the world, humans had only 23 pairs of chromosomes. But how did we even get to know this? The simple answer is we counted our chromosomes through a process called karyotyping. And karyotyping is basically a laboratory technique used to examine the number, size, and structure of chromosomes in a cell. But how does it work? The process begins with collecting a sample of cells, often from blood, and culturing them to promote cell division. Cell division is basically how the cell multiplies and is the only time we can actually count the chromosomes. This is because it is the only time when chromosomes are fully condensed and distinct enough to be counted accurately. See, during cell division, particularly in the metaphase stage of mitosis, cell division results in two daughter cells, each with the same number of chromosomes as the original cell. The chromosomes are tightly coiled and visible under a microscope. It is during this phase that the chromosomes line up at the cell center, making them more accessible and distinguishable for counting outside of cell division. Chromosomes are less organized and spread out as chromat, which makes them difficult to count and analyze. Therefore, by examining cells during mitosis, scientists can successfully count and study chromosomes to detect abnormalities and understand genetic information. To view chromosomes clearly, the cells are treated with a chemical to stop the division during metaphase. After harvesting and breaking open the cells, the chromosomes are stained with specific dyes that create distinctive banding patterns. These stained chromosomes are spread on a microscope slide and analyzed under a microscope. It is then that the chromosomes are arranged into a karyotype, which is a standardized format that organizes them into pairs based on size, shape, and banding. This arrangement allows for easy identification of any abnormalities, such as extra or missing chromosomes and structural changes. By examining the karyotype, scientists can diagnose genetic conditions, study chromosomal abnormalities, and in the case of the Human Genome Project, discover how we lost a chromosome. But what happened to our 24th chromosome? Here's the thing. The chromosome of any species is so important that if a single one were to be lost in an individual, that individual runs a high chance of mortality and most likely wouldn't make it past infancy. So what makes us the exception? The simple answer is we aren't. We didn't lose a chromosome, but something amazing did happen. In 2005, a study was published that showed the human chromosome and its correspondence in chimps. As you would expect, our chromosome 1 aligned with the chimpanzee's chromosome 1. This means they had similar size, shape, and banding. However, our chromosome 2 was where things got unusual. There was no corresponding chromosome 2 in the chimps. To make this more confusing, our chromosome 3 was there 2, our 4 was there 3, and so on. This pattern once again fell apart when we began looking at chromosomes 13 and 14 of apes. Now this time. However, it was we who had no corresponding chromosome. Our chromosome 13 was their chromosome 14, and so on. So, what was going on? Where was their chromosome 2? And why do we not have a corresponding chromosome for their 13 and 14? This was the question scientists asked, and to find the answer, they looked at our weirdest chromosome, chromosome 2. It was here they found something groundbreaking. Normally, the chromosome has these two markers named the telome and the centromere. The telome and centromere are essential components of chromosomes, each playing a unique role in maintaining chromosome stability and function. The telome are repetitive DNA sequences located at the ends of chromosomes. They act as protective caps, preventing the chromosome ends from deteriorating or fusing with other chromosomes. This protective function ensures that vital genetic information is preserved during cell division. And essentially, each time a cell divides, the telome shortens slightly, contributing to cellular aging. When they become too short, this can lead to cellular issues. Centromeres, on the other hand, are specialized regions located at the center of chromosomes, where the two sister chromatids are joined. They play a critical role during cell division by ensuring the chromosomes are accurately segregated into daughter cells. The centromere also serves as the attachment point for the kinetochore, a protein structure that links the chromosomes to the spindle fibers, which pull the chromosomes apart. Proper function of the centromere is essential for chromosome stability, and any abnormalities can lead to chromosome segregation, potentially causing genetic disorders or cancers. This was what made chromosome 2 unique. Upon examination, scientists found that it had telomeres in the middle where they shouldn't be. But that wasn't all, as it also had one centromere in the middle. Further along the section, there was an extremely similar DNA code to what you would expect from another centromere. Essentially, there was some DNA code resembling a telomere code located between two centromeres. This discovery changed the way we look at our evolution forever, so much so that it even caused the renumbering of the ape chromosome to line up with the human chromosome. But how exactly did chromosome 2 affect us as humans? Quick pause. If you're enjoying this journey through the prehistoric world, don't forget to like and subscribe. More than 97% of our viewers watch without subscribing, and we'd love to have you join our tribe. It would make all the difference, so if it's done, great. Thanks.